Human evolution and the importance of studying human historical and cultural heritage. The evolution of humans is a fascinating journey that spans millions of years. Throughout this evolutionary process, humans have developed complex societies, languages, technologies, and cultural practices that have shaped our history and identity. The study of human cultural and historical heritage plays a crucial role in understanding our origins, development, and the challenges we face in the modern world. One of the primary reasons why researching human cultural and historical heritage is significant is that it provides insights into our roots and the path we have taken as a species. By studying artifacts, archaeological sites, ancient texts, and oral traditions, researchers can reconstruct the story of our ancestors and trace the evolution of our civilizations. This knowledge allows us to connect with our past, appreciate our shared human heritage, and gain a deeper understanding of the diverse cultures and societies that have shaped our present. Cultural and historical heritage also serves as a foundation for identity and a source of collective memory. It helps us understand who we are as individuals and as communities, providing a sense of belonging and continuity across generations. By exploring our cultural and historical heritage, we can uncover shared values, beliefs, and experiences that bind us together and foster social cohesion. This understanding of our collective identity can promote empathy, tolerance, and appreciation for the diversity of human cultures. Moreover, studying cultural and historical heritage provides valuable lessons and insights that can inform our present and future decisions. By examining the successes, failures, and innovations of past societies, we can learn from their experiences and apply their wisdom to address contemporary challenges. Understanding how different cultures have adapted to environmental changes, managed resources, and resolved conflicts can inspire sustainable practices, foster intercultural dialogue, and contribute to the development of inclusive and equitable societies. Cultural heritage also holds economic value and can contribute to the well-being of communities. Historic sites, museums, and cultural tourism can generate income, create job opportunities, and stimulate local economies. Preserving and promoting cultural heritage can enhance the quality of life for communities by celebrating their unique identity, attracting visitors, and fostering cultural exchange and dialogue. Furthermore, the study of human cultural and historical heritage provides opportunities for interdisciplinary research and collaboration. Scholars from various fields, including archaeology, anthropology, history, linguistics, and sociology, come together to unravel the mysteries of the past and gain comprehensive insights into human evolution. This interdisciplinary approach fosters innovation, encourages critical thinking, and promotes a holistic understanding of the complexities of human societies. Preserving and protecting cultural and historical heritage is of paramount importance. As we advance technologically and face the challenges of globalization, cultural heritage is at risk of being lost or eroded. Rapid urbanization, environmental degradation, armed conflicts, and illicit trafficking of cultural artifacts pose significant threats to our shared human heritage. Therefore, efforts to safeguard and conserve cultural and historical sites, artifacts, and intangible heritage are crucial for future generations. Continuing the exploration of the significance of studying human cultural and historical heritage, it is important to emphasize the role it plays in promoting intercultural understanding and fostering dialogue among diverse communities. Cultural heritage provides a platform for sharing and exchanging ideas, beliefs, and traditions, facilitating a deeper appreciation and respect for different cultures. By studying the cultural and historical heritage of different societies, we gain insight into the experiences, struggles, and achievements of past generations. This knowledge helps us recognize the commonalities that unite us as human beings, transcending geographic and cultural boundaries. It promotes empathy, tolerance, and a sense of global citizenship by fostering an understanding of the shared aspirations and challenges faced by humanity throughout history. Furthermore, the study of cultural heritage enables us to address contemporary issues and shape a more sustainable future. Many of the challenges we face today, such as climate change, social inequality, and cultural preservation, can benefit from the lessons learned from our cultural and historical heritage. By examining the ways in which past societies adapted to environmental changes, managed resources, and built resilient communities, we can gain valuable insights into strategies for addressing these pressing issues in the present. Cultural and historical heritage also has the power to inspire creativity, innovation, and cultural expression. It provides a rich source of inspiration for artists, writers, filmmakers, and other creative individuals who draw upon the stories, symbols, and traditions of the past to create meaningful works of art. Cultural heritage serves as a wellspring of ideas and narratives that shape contemporary cultural production and contribute to the enrichment of our collective human experience. Moreover, the preservation and study of cultural heritage contribute to the field of education. By incorporating cultural and historical heritage into educational curricula, we can instill a sense of pride in students' cultural identities, promote cultural diversity, and foster critical thinking skills. Through interactive learning experiences, students can engage with their cultural heritage, develop a deeper understanding of their own identities, and become active participants in preserving and promoting cultural heritage for future generations. Cultural heritage also plays a vital role in tourism and economic development. Historic sites, museums, and cultural events attract tourists from around the world, generating economic opportunities and supporting local communities. The preservation and promotion of cultural heritage can stimulate job creation, entrepreneurship, and sustainable tourism practices that respect local traditions and natural environments. Continuing the exploration of the significance of studying human cultural and historical heritage, it is important to highlight the role it plays in fostering a sense of identity and belonging among individuals and communities. Cultural heritage provides a link to our past and allows us to understand the values, traditions, and beliefs that have shaped us as a society. By studying our cultural heritage, we can develop a deeper sense of self-awareness, strengthen our cultural identity, and build a sense of belonging within our communities. Cultural and historical heritage also serves as a source of inspiration and innovation. By examining the achievements and contributions of past civilizations, we can learn from their successes and failures, and apply that knowledge to drive progress and innovation in various fields. The study of cultural heritage can inspire new ideas, spark creativity, and lead to the development of new technologies, art forms, and societal advancements. Additionally, cultural heritage holds immense educational value. It provides a unique opportunity for individuals, especially the younger generation, to learn about their roots and gain a comprehensive understanding of their cultural heritage. By studying the cultural and historical context of their communities, young people can develop a sense of pride and appreciation for their heritage, fostering a stronger connection to their roots and a deeper respect for cultural diversity. The preservation and conservation of cultural heritage also contribute to the economic growth and sustainable development of communities. Global distribution of modern humans and the development of cultures and languages. The global distribution of modern humans is a testament to our remarkable ability to adapt and thrive in diverse environments. Today, human populations are spread across the globe, exhibiting a rich tapestry of cultural diversity, linguistic variation, and unique social structures. The study of human distribution and the development of cultures and languages provides valuable insights into the complex interactions between societies and their environments. The distribution of modern humans is shaped by a variety of factors, including historical migrations, geographic features, resource availability, and social and political dynamics. Throughout history, humans have migrated and settled in different regions, spreading their cultural practices, traditions, and languages. From the earliest migrations out of Africa to the exploration and colonization of new lands, human movement has shaped the global distribution of populations and influenced the development of diverse cultures. The development of cultures and languages is closely intertwined with human adaptation to local environments. Different ecological contexts, such as arid deserts, dense rainforests, or high-altitude mountains, have shaped unique cultural practices and technologies. People have developed specialized knowledge and skills to survive and thrive in specific environments, from nomadic herding in arid grasslands to sustainable agriculture in fertile river valleys. These adaptations have given rise to distinct cultural practices, traditions, and ways of life. Language is a fundamental aspect of human culture and plays a crucial role in shaping social identity and communication. The development of languages is closely tied to the historical, cultural, and geographic context in which they emerged. Languages reflect the experiences, values, and beliefs of their speakers, providing a rich tapestry of linguistic diversity. They are not only a means of communication but also carriers of cultural heritage and repositories of collective knowledge. The coexistence of different languages and cultures within a globalized world presents opportunities for cultural exchange, collaboration, and mutual understanding. Multilingualism fosters cross-cultural communication, promotes tolerance and respect, and opens doors to diverse perspectives and ideas. It enriches human interactions, enhances intercultural dialogue, and contributes to the development of innovative solutions to global challenges. However, the distribution and development of cultures and languages are not without challenges. Globalization, urbanization, and technological advancements have led to the homogenization of cultures and the erosion of linguistic diversity. The dominance of a few major languages and cultural influences poses a threat to the survival of smaller languages and indigenous cultures. Preserving and revitalizing endangered languages and cultural practices is crucial for maintaining the richness and diversity of our global heritage. Continuing the exploration of the global distribution of modern humans and the development of cultures and languages, it is important to acknowledge the dynamic nature of these processes and the various factors that contribute to their evolution and growth. The global distribution of modern humans is the result of centuries of migration, colonization, and globalization. Throughout history, humans have embarked on journeys in search of better opportunities, resources, or refuge, leading to the establishment of diverse populations in different parts of the world. Factors such as economic opportunities, political stability, and social networks have influenced the patterns of human migration and settlement. The development of cultures and languages is intricately linked to human interactions, social dynamics, and historical events. Cultures evolve through the accumulation of shared knowledge, beliefs, values, and practices within a particular group or society. They are shaped by historical events, including conquests, trade networks, and cultural exchanges. The development of languages follows a similar trajectory, as they adapt to meet the needs of communication and reflect the cultural and social contexts in which they emerge. The growth and evolution of cultures and languages are also influenced by factors such as environmental conditions, technological advancements, and social changes. Environmental factors, such as the availability of resources and climate conditions, can shape the economic activities and lifestyles of communities, giving rise to distinct cultural practices and adaptations. Technological advancements, such as the invention of writing systems or the development of communication technologies, have had a profound impact on the transmission and preservation of languages and cultural knowledge. Migration, both forced and voluntary, has played a significant role in the spread and development of cultures and languages. When different communities come into contact through migration, trade, or conquest, they often exchange ideas, customs, and languages, leading to cultural blending, syncretism, and linguistic borrowing. This cultural and linguistic diversity is a reflection of the interconnectedness and complexity of human societies. The development and preservation of cultures and languages are essential for maintaining social cohesion, identity, and intergenerational transmission of knowledge. They contribute to the richness of human experiences, creativity, and innovation. Cultural and linguistic diversity also fosters mutual understanding, respect, and appreciation among different communities, promoting dialogue and cooperation. However, the rapid globalization and interconnectedness of the modern world pose both opportunities and challenges for the development and preservation of cultures and languages. Globalization brings increased access to information, intercultural exchange, and economic opportunities, but it also leads to the dominance of certain cultural and linguistic influences, potentially marginalizing or eroding local traditions and languages. Continuing the exploration of the global distribution of modern humans and the development of cultures and languages, it is important to recognize the significant impact of cultural exchange. Globalization and technological advancements on the evolution and interconnectedness of societies worldwide. The global distribution of modern humans reflects the intricate web of historical events, migrations, and interactions between different groups and regions. Human populations have migrated and settled in various parts of the world, adapting to local environments, resources, and social dynamics. As a result, diverse cultural traditions, customs, and languages have emerged, each with its unique identity and contributions to the global tapestry of human heritage. The development of cultures and languages is not a static process but rather a dynamic and ongoing phenomenon. Cultures evolve and transform in response to social, economic, and environmental changes. Technological advancements, such as the Internet, social media, and global communication networks, have facilitated the exchange of ideas, knowledge, and cultural practices, leading to the emergence of hybrid cultures and the blending of traditions. In the modern interconnected world, the growth and development of cultures and languages are shaped by globalization. Economic globalization has led to increased trade, travel, and cultural exchange, fostering the dissemination of cultural products, ideas, and values across borders. This has created opportunities for cultural enrichment, cross-cultural collaborations, and the celebration of diversity. However, globalization also presents challenges, as dominant cultural forces can lead to the marginalization of local cultures and the loss of linguistic diversity. Rare plant species and how to protect and conserve them to ensure their viability. Rare plant species play a crucial role in maintaining biodiversity and ecological balance. However, they face numerous threats that put their survival at risk. It is imperative to implement effective strategies for their protection and conservation to ensure their long-term survival and the preservation of our natural heritage. Rare plant species are characterized by their limited distribution and small population sizes, making them particularly vulnerable to human activities, habitat loss, climate change, and invasive species. These species often possess unique genetic traits and ecological functions that contribute to ecosystem resilience and provide valuable ecosystem services. Therefore, their conservation is vital for maintaining overall ecosystem health and functionality. One approach to protecting and conserving rare plant species is through the establishment of protected areas. These designated zones provide legal safeguards and management measures to ensure the preservation of critical habitats and ecosystems. Protected areas not only offer a sanctuary for rare plant species but also promote the conservation of associated flora and fauna, creating a network of interconnected habitats that support biodiversity conservation on a larger scale. In addition to protected areas, ex situ conservation measures are also employed to safeguard rare plant species. This involves the collection and storage of seeds, spores, or living plant material in specialized facilities, such as botanical gardens, seed banks, or tissue culture laboratories. These repositories serve as a backup in case of catastrophic events or habitat loss, and they also provide opportunities for research, education, and the potential reintroduction of species into the wild. Collaborative efforts between governments, conservation organizations, and local communities are essential for effective conservation. The involvement of local communities is particularly important as they often have traditional knowledge and practices that can contribute to the sustainable management and conservation of rare plant species. Engaging communities in conservation initiatives not only enhances local ownership and stewardship but also promotes the equitable sharing of benefits and the preservation of cultural values associated with these plants. Furthermore, raising awareness and promoting public education about the importance of rare plant species is crucial. By fostering a sense of environmental stewardship and highlighting the value of biodiversity, individuals and communities can become active participants in conservation efforts. Educational programs, outreach activities, and citizen science initiatives can empower people to contribute to monitoring, habitat restoration, and the protection of rare plant species in their local environments. Policy interventions and legal frameworks are instrumental in ensuring the conservation of rare plant species. Governments can establish laws and regulations to protect critical habitats, restrict unsustainable practices, and promote sustainable land use planning. International agreements, such as the Convention on Biological Diversity, provide a global framework for biodiversity conservation and encourage collaboration among nations in addressing the conservation needs of rare plant species. Furthermore, research and monitoring are fundamental in guiding conservation efforts. Scientific studies can provide valuable insights into the ecological requirements, population dynamics, and threats facing rare plant species. By understanding their specific needs and vulnerabilities, conservation strategies can be tailored to address these challenges effectively. Continuing the discussion on the protection and conservation of rare plant species, it is essential to address the various challenges and implement innovative approaches to ensure their long-term survival and sustainable management. One of the key challenges in conserving rare plant species is habitat loss and degradation. Human activities such as deforestation, urbanization, and agriculture encroach upon natural habitats, leading to the destruction of critical ecosystems where rare plants thrive. To combat this, habitat restoration initiatives can be implemented, including reforestation efforts, wetland rehabilitation, and the establishment of protected areas to create safe havens for these plants. Invasive species also pose a significant threat to rare plant species. Invasive plants can outcompete native species for resources and disrupt the delicate balance of ecosystems. Effective management strategies, including early detection, rapid response, and control measures, are necessary to mitigate the impacts of invasive species on rare plant populations. Additionally, public awareness campaigns can educate communities about the dangers of invasive species and encourage responsible practices in gardening and landscaping. Climate change presents another major challenge for the conservation of rare plant species. Alterations in temperature, precipitation patterns, and extreme weather events can disrupt the natural habitats and life cycles of these plants. To address this, conservation efforts should include strategies for climate change adaptation, such as assisted migration, assisted evolution, and the promotion of resilient habitats that can withstand changing climatic conditions. Collaboration with climate scientists and utilizing predictive models can aid in identifying areas of high conservation priority and developing targeted conservation plans. In situ conservation, which involves protecting rare plant species within their natural habitats, is a crucial approach. It involves the establishment of protected areas, the enforcement of regulations to prevent illegal harvesting or habitat destruction, and the promotion of sustainable land use practices. Engaging local communities and indigenous peoples in conservation efforts is vital, as they possess traditional knowledge and practices that can contribute to the effective management and preservation of rare plants. Ex situ conservation, such as seed banking and botanical gardens, also plays a critical role in safeguarding rare plant species. Seeds are collected and stored in seed banks, ensuring their long-term viability and availability for future restoration efforts. Botanical gardens serve as living repositories, maintaining collections of rare plants and conducting research on their biology and conservation. These ex situ measures provide an additional layer of protection and serve as a valuable resource for research, education, and public engagement. International collaboration and cooperation are essential for the conservation of rare plant species. Sharing knowledge, expertise, and resources among countries can enhance conservation efforts and promote the exchange of best practices. International agreements and conventions, such as the International Union for Conservation of Nature, UCAN, Red List, facilitate the assessment of the conservation status of rare plants and guide conservation priorities at the global level. Lastly, public engagement and education are critical for the success of conservation efforts. Raising awareness about the value and importance of rare plant species can foster a sense of responsibility and inspire individuals to take action. Cell diversity, prokaryotic versus eukaryotic cells. Every living organism is made up of cells that are either prokaryotic or eukaryotic. Both types of cells have essential features that enable living organisms to perform their biological functions, but they differ in structure, function, and complexity. The diversity of cells is a fascinating area of study, and this article aims to explore the differences between prokaryotic and eukaryotic cells. Prokaryotic cells. Prokaryotic cells are the simplest and most primitive form of cells that exist in nature. They are found in bacteria and archaea and are unicellular organisms. Prokaryotic cells are characterized by their small size, lack of nucleus, and absence of membrane-bound organelles. Instead, their genetic material is scattered in the cytoplasm of the cell. The plasma membrane is the outermost layer of the prokaryotic cell, and it serves as a barrier between the internal environment of the cell and the external environment. The cytoplasm is a gel-like substance that fills the cell, and it contains various cellular components, including the genetic material, ribosomes, and enzymes. Ribosomes are responsible for protein synthesis in prokaryotic cells. One of the critical features of prokaryotic cells is their cell walls. The cell wall serves as a protective layer that helps the cell withstand external pressures and maintain its shape. In bacteria, the cell wall is made up of peptidoglycan, a complex molecule consisting of sugars and amino acids. Archaea, on the other hand, have cell walls made up of different materials and chemical structures. Another unique feature of prokaryotic cells is the presence of flagella. Flagella are long, whip-like structures that protrude from the cell wall and enable the cell to move. The movement of flagella is driven by the rotation of a motor-like structure located in the cell membrane. Prokaryotic cells reproduce by a process known as binary fission. During binary fission, the genetic material replicates, and the cell divides into two identical daughter cells. This process is relatively simple and fast, allowing prokaryotic cells to multiply rapidly and survive in different environments. Eukaryotic cells. Eukaryotic cells are more complex and evolved compared to prokaryotic cells. They are found in plants, animals, fungi, and protists, and they are larger in size than prokaryotic cells. Eukaryotic cells have a well-defined nucleus, which contains the genetic material and is separated from the cytoplasm by a double-membrane nuclear envelope. The cytoplasm of eukaryotic cells is more complex than that of prokaryotic cells. It contains membrane-bound organelles, including mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, and peroxisomes. Each of these organelles has a specific function and plays a crucial role in the overall cellular functioning. Mitochondria are the powerhouse of the eukaryotic cell. They are responsible for generating energy in the form of ATP through cellular respiration. The endoplasmic reticulum is involved in protein synthesis and transport within the cell. The Golgi apparatus is responsible for sorting, modifying, and packaging proteins that are secreted out of the cell. Lysosomes are involved in breaking down waste material and cellular debris, while peroxisomes are responsible for breaking down toxic substances. The cytoskeleton is another significant component of eukaryotic cells. It is a network of protein fibers that provides structural support to the cell and enables movement. It consists of three types of fibers: actin filaments, microtubules, and intermediate filaments. Actin filaments are responsible for cell movement and maintaining the shape of the cell. Microtubules are involved in cell division, intracellular transport, and movement of cilia and flagella. Intermediate filaments provide mechanical support to the cell and help maintain its shape. Eukaryotic cells undergo a more complicated process of cell division known as mitosis. During mitosis, the genetic material duplicates, and the cell divides into two identical daughter cells. The process of mitosis involves several stages, including prophase, metaphase, anaphase, and telophase, each of which is critical for the accurate distribution of genetic material. Furthermore, the study of cells has allowed us to develop various medical treatments and therapies that have benefited millions of individuals worldwide. For example, the study of eukaryotic cells has helped researchers understand how cancer develops and spreads, leading to the development of targeted therapies that have saved countless lives. The study of prokaryotic cells has also led to the development of antibiotics that can treat bacterial infections. In addition to medical applications, the study of cells has also had significant implications in fields such as biotechnology and agriculture. The ability to manipulate and engineer cells has led to the development of genetically modified organisms that are more resistant to pests and have higher yields. This technology has helped increase food production and reduce hunger worldwide, benefiting individuals and societies alike. Finally, understanding the differences between prokaryotic and eukaryotic cells has important implications for the study of evolution. The presence of distinct and diverse cell types is a testament to the complexity and diversity of life on Earth. It demonstrates how life has evolved and adapted to various environments, and reinforces the concept of natural selection and survival of the fittest. In the future, we may be able to use our knowledge of cells to develop even more advanced medical treatments and therapies. For example, stem cell research has already shown promising results in treating various diseases and conditions, such as Parkinson's disease, diabetes, and spinal cord injuries. Additionally, the use of gene editing technology, such as CRISPR-Cas9, may allow us to correct genetic mutations that cause inherited diseases and disorders. Moreover, the study of cells has also led to the development of new diagnostic tools that allow doctors and researchers to detect, diagnose, and monitor diseases more accurately and effectively. For example, various types of cellular assays can measure the presence and activity of specific cells, proteins, or genes, providing insights into disease progression and treatment efficacy. The implications of cell research extend beyond human health and into the realm of environmental science and sustainability. Understanding how cells interact with their environment can help us develop more sustainable practices in agriculture, energy production, and waste management. For example, understanding how bacteria and other microorganisms decompose organic waste can lead to the development of more effective and efficient composting systems. While there is still so much to be discovered and learned about cells, one thing is certain: the study of cells will continue to be a crucial area of research in the coming years. From understanding the basic building blocks of life to developing innovative treatments and therapies for diseases, the applications of cell research are vast and highly beneficial. As we develop a deeper understanding of cells, we will be better equipped to address some of the world's most pressing challenges, from climate change and environmental degradation to complex diseases and global epidemics. The insights we gain from studying cells and their behavior will allow us to devise more effective strategies for addressing these challenges and promoting a healthier and more sustainable future for all. Ultimately, the study of cells underscores the interconnectedness of all living things on Earth. By unraveling the mysteries of cells and how they function, we can gain a deeper appreciation for the complexity and diversity of life, and the ways in which we are all interconnected as part of a larger, intricate, and constantly evolving ecosystem. As we strive towards a more comprehensive and interdisciplinary approach to science, the study of cells will remain an integral part of our understanding of the natural world. By combining insights from different fields, we can unlock new insights and develop innovative solutions to some of the world's most pressing challenges. For example, the recent COVID-19 pandemic has highlighted the critical role that cell studies play in the development of vaccines and treatments. Understanding the cellular mechanisms by which the virus infects and spreads within the body has allowed researchers to develop targeted treatments and vaccines that have already saved countless lives. From DNA to protein. Cell division, mitosis versus meiosis. Cell division, mitosis versus meiosis. Cell division is a critical process in the growth and reproduction of organisms. It is a process whereby a cell divides into two or more cells. In higher organisms, cell division is essential for growth, repair, and reproduction. There are two types of cell division, mitosis and meiosis. In this article, we will delve into the differences between mitosis and meiosis and their importance in biological processes. Mitosis is the process of cell division whereby a parent cell divides into two daughter cells that are genetically identical to the parent cell. The process is a continuous series of events starting from the growth phase, G1, to synthesis phase, S, growth phase, G2, and finally mitosis phase. In the growth phase, G1, a cell increases its size and prepares itself for DNA replication. In the synthesis phase, S, in the growth phase, G2, the cell prepares for mitosis. In the mitosis phase, the duplicated chromosomes separate into two identical nuclei, each containing the same number of chromosomes as the parent cell. Mitosis is a fundamental process in the growth and repair of tissues in multicellular organisms. It ensures that daughter cells have the same genetic material as the parent cell. During development, mitosis is essential for the growth and the formation of new organs and tissues. After injury, mitosis aids in cell regeneration, allowing tissues to heal and recover. Mitosis is also crucial in asexual reproduction, where organisms can generate offspring without the need for a mate. On the other hand, meiosis is the process of cell division whereby a single cell divides into four genetically diverse daughter cells. The process is also a series of events, starting with the growth phase, G1, synthesis phase, S, growth phase, G2, and meiosis I and II phase. In the growth phase, G1, the cell increases its size and prepares for DNA replication. During the synthesis phase, S, in the growth phase, G2, the cell prepares for the first round of meiosis. During meiosis I, homologous chromosomes pair and exchange genetic material through a process called crossing over. This process results in genetically diverse gametes. In meiosis II, the sister chromatids of each chromosome are separated, resulting in four genetically diverse daughter cells. Meiosis is the process of cell division that is essential for sexual reproduction in eukaryotes. A significant difference between mitosis and meiosis is the number of daughter cells produced. Mitosis produces two genetically identical daughter cells, while meiosis produces four genetically diverse daughter cells. Additionally, mitosis occurs in somatic cells, while meiosis occurs in sex cells, gametes. Another critical difference is the genetic diversity produced by the two processes. Mitosis produces genetically identical daughter cells, while meiosis produces genetically diverse daughter cells. By shuffling genetic material between homologous chromosomes and separating sister chromatids, meiosis produces daughter cells containing various combinations of genetic material from both parents. This genetic diversity increases the chances of survival in changing or challenging environments and contributes to an organism's adaptability. Mitosis and meiosis both begin with the same process of DNA replication during the synthesis phase, S. However, during meiosis, crossing over can occur, which does not take place in mitosis. During crossing over, homologous chromosomes exchange genetic material, resulting in genetic diversity. This process does not occur in mitosis, which only duplicates chromosomes without introducing genetic variation. While mitosis is essential for growth and repair, meiosis is essential for sexual reproduction. During sexual reproduction, fertilization combines the genetic material of two parents, resulting in genetically diverse offspring. Meiosis also produces gametes, which are necessary for fertilization. Scientists continue to study the mechanisms that govern cell division and the regulation of these processes. This research has led to the development of new techniques for diagnosis, therapeutic interventions, and gene therapies. It has also allowed us to understand more about the genetic origins of diseases that result from errors in cell division, like Down syndrome or sickle cell anemia. Furthermore, understanding the differences between mitosis and meiosis has practical applications in plant and animal breeding programs. These programs aim to select and propagate organisms with desirable genetic traits, such as increased yield or resistance to disease. By understanding the mechanisms of meiosis and breeding individuals with desired traits, breeders can produce new generations of genetically diverse plants and animals. Scientists are also studying how environmental factors can impact cell division. Research shows that environmental stressors such as exposure to toxins, radiation, or changes in temperature can affect the frequency of chromosome aberrations, which can cause genetic disorders or cancer. Understanding how these factors impact cell division can help us mitigate their effects and develop preventative measures to protect human health. Moreover, advances in technology have allowed scientists to study cell division on a molecular level, providing new insights into the complex mechanisms that govern these processes. For example, researchers are investigating the roles of various proteins and enzymes involved in cell division, which could lead to the development of new therapies for diseases like cancer. In addition, research has shown that some organisms have evolved unique methods of cell division that differ from the typical mitosis or meiosis processes. For example, some bacteria have developed binary fission, a process in which the parent cell splits into two identical daughter cells. Other organisms, such as certain fungi and algae, undergo a type of cell division called mitosis without cytokinesis, in which the nucleus divides but the cell does not fully split into two cells. Cell division also plays a crucial role in the development of multicellular organisms. During embryonic development, cells divide and differentiate into various cell types, each with specific functions. In some cases, cells can differentiate and revert to an earlier stage of development, allowing them to repair or regenerate damaged tissues. This process is known as cellular reprogramming and has implications for regenerative medicine and tissue engineering. Regenerative medicine aims to restore damaged or diseased tissues by using cellular reprogramming techniques to generate new cells or tissues. For example, scientists have used induced pluripotent stem cells, IPSCs, to generate new cardiac cells and tissues for patients with heart disease. IPSCs are created by reprogramming adult cells to an embryonic-like state, allowing them to differentiate into various cell types. In tissue engineering, researchers use a combination of cells, scaffolds, and bioactive molecules to create functional tissues or organs outside of the body. By understanding the mechanisms of cell division and differentiation, scientists can design more effective tissue engineering strategies to replace or repair damaged tissues or organs. In addition, research has shown that cell division can be influenced by external factors, such as growth factors, hormones, and mechanical forces. For example, mechanical stress can modulate the rate and direction of cell division, affecting tissue growth and repair. By understanding the effects of these external factors on cell division, scientists can develop new strategies to promote tissue regeneration and repair. In addition, researchers are exploring the potential of targeting the cell division process in cancer treatment. Cancer cells divide uncontrollably, leading to the formation of tumors. Targeting the mechanisms of cell division could be an effective strategy to halt the growth and spread of cancer cells. For example, some chemotherapy drugs work by inhibiting cell division, preventing cancer cells from replicating and growing. Moreover, researchers are studying the role of telomeres in cell division and aging. Telomeres are the caps at the ends of chromosomes that protect them from deteriorating or fusing with other chromosomes. Each time a cell divides, its telomeres become shorter, ultimately leading to cell death. This process is thought to contribute to the aging process and age-related diseases. Recent research has shown that telomere shortening can be prevented or slowed down by telomerase, an enzyme that adds DNA to the ends of telomeres. Cell signaling, communication between cells. Cell signaling is a vital process by which cells communicate with each other to carry out essential functions such as growth, development, and response to environmental stimuli. It involves the transmission of molecular signals between different cells, which allows them to coordinate and integrate their activities to maintain homeostasis in organisms. It is a complex process that is crucial for the proper functioning of cells and organisms. The basics of cell signaling. Cell signaling involves the transmission of information between cells via different signaling pathways. Typically, cells release signaling molecules or ligands that bind to receptors on target cells, triggering a cascade of events that ultimately lead to a cellular response. The cellular response can be anything from the activation of specific metabolic pathways to changes in gene expression or cell division. The different types of signaling pathways can be broadly classified into three categories based on the distance between the cells involved. These include endocrine, paracrine, and autocrine signaling. Endocrine signaling. It occurs when hormones are released into the bloodstream that travels to different parts of the body to affect target cells. Hormones are signaling molecules that are produced by specialized cells and released into the bloodstream. They are then carried to their target organs where they bind to specific receptors, triggering a cellular response. Paracrine signaling. It occurs when molecules are released by a cell that affects the neighboring cells. The affected cells are generally in close proximity to the signaling cell. One of the best examples of paracrine signaling is the interaction between neurons, which rely on neurotransmitters to transmit signals between cells. Autocrine signaling. It is a type of paracrine signaling where the signaling cell and the target cell are the same cell type. This occurs when a cell produces signaling molecules that bind to receptors on its surface, leading to cellular responses. Examples include cancer cells and some immune cells that rely on autocrine signaling to maintain their growth and function. The signaling molecules involved in cell signaling. The signaling molecules or ligands involved in cell signaling are of different types and are synthesized by cells of various tissues and organs. Generally, the molecules involved in signaling fall into four broad categories. 1. Small molecules. These are small organic molecules consisting of complex chains of atoms. Examples of small molecules include neurotransmitters, hormones, gas, and growth factors. 2. Peptides and proteins. These are larger molecules consisting of long chains of amino acids that form complex 3D structures. Examples include insulin, cytokines, and growth factors. 3. Lipids. These are hydrophobic molecules that include steroid hormones, prostaglandins, and fatty acid derivatives. They are primarily involved in paracrine and autocrine signaling. 4. Nucleotides. These include molecules such as cyclic AMP, CAMP, which is involved in the signaling pathway triggered by G-protein coupled receptors, GPCRS. The receptors involved in cell signaling. The signaling molecules released by the cells bind to specific receptors located on the surface of the target cells. The receptors can be classified into two groups based on their location: surface receptors and intracellular receptors. Surface receptors. These are membrane-bound receptors that are located on the surface of the target cells. Different types of surface receptors include ion channels, G-protein coupled receptors, GPCRS, and receptor tyrosine kinases, RTKS. Intracellular receptors. These receptors are located inside the cell and are activated by signaling molecules that can pass through the plasma membrane. Examples of intracellular receptors include steroid hormone receptors, thyroid hormone receptors, and retinoid receptors. The signaling pathways involved in cell signaling. There are several different types of signaling pathways involved in cell signaling. These include 1. Ion channel receptors. These are membrane-bound proteins that form channels in the plasma membrane through which ions can pass. When a signaling molecule binds to these receptors, the channels open, allowing ions to pass through, which leads to changes in membrane potential and cellular responses. 2. G-protein coupled receptors. These receptors are the most common type of receptors and are involved in several signaling pathways. When a signaling molecule binds to these receptors, it activates a G-protein that triggers a cascade of events, ultimately leading to cellular responses. 3. Receptor tyrosine kinases. These are a type of surface receptor that activates intracellular signals by phosphorylating tyrosine residues. The signaling leads to downstream activation of various pathways involved in cellular growth and differentiation. 4. Intracellular receptors. As the name suggests, these receptors are located inside the cell and are activated by signaling molecules that can pass through the plasma membrane. The intracellular signaling pathways are primarily involved in transcriptional regulation. The importance of cell signaling in health and disease. Cell signaling is crucial for maintaining homeostasis in organisms. Any defects or issues in the signaling pathways can lead to a wide variety of diseases, including cancer, autoimmune disorders, and hormonal disorders. Cancer, for example, is caused by the activation of oncogenic signaling pathways that promote uncontrolled growth and proliferation of cells. Similarly, immune system disorders occur when the immune cells fail to signal effectively to mount an appropriate response against invading pathogens. Scientists continue to study and explore cell signaling to gain a better understanding of its mechanisms and pathways. One area of research is the role of cell signaling in aging and age-related diseases. As we age, the signaling pathways become less efficient, leading to functional declines in various organs and tissues. Identifying ways to promote cellular communication and enhance signaling pathways could potentially delay the onset of age-related diseases and improve overall health in aging populations. Furthermore, researchers are also investigating how to use the principles of cell signaling to develop new therapies for various diseases. For example, drug development companies are creating drugs that target specific receptors or enzymes involved in signaling pathways to treat cancer, autoimmune disorders, and metabolic diseases. These drugs can block or enhance signaling pathways to alter cellular responses and prevent or treat diseases. As our understanding of cell signaling continues to grow, new technologies are being developed to study this process more effectively. For example, fluorescence microscopy allows researchers to visualize the dynamic movements of signaling molecules within cells in real time. Additionally, techniques like optogenetics enable researchers to use light to control the activity of specific signaling molecules or cells, providing a powerful tool for understanding the role of different signaling pathways in cellular function and disease. The study of cell signaling is an exciting and rapidly evolving field with vast potential for improving our understanding of health and disease. Cellular senescence, the science of aging. As we age, our bodies undergo changes that are both visible and invisible. We develop wrinkles, our hair thins out, and our bones become frail. Many of these changes are thought to be the result of a process known as cellular senescence. Cellular senescence is a normal part of aging, but it can also contribute to the development of age-related diseases, such as cancer, cardiovascular disease, and Alzheimer's disease. What is cellular senescence? Cellular senescence is essentially a state of programmed cell growth arrest that happens as we age. When a cell becomes senescent, it stops dividing and can no longer perform its usual functions. Instead, it becomes metabolically active and secretes a range of molecules known as the senescence-associated secretory phenotype, SASP. The SASP includes a wide array of molecules, including cytokines, growth factors, and other signaling molecules that can have both positive and negative effects on the body. Although cellular senescence is a normal part of aging, it can also be triggered by a variety of other circumstances, including DNA damage, telomere shortening, oxidative stress, and inflammation. These triggers can cause cells to become senescent prematurely, and can also contribute to the development of age-related diseases. The impact of cellular senescence on aging. Although cellular senescence is a normal part of aging, it can also contribute to the development of many age-related diseases. One reason for this is that the SASP can have both positive and negative effects on the body. For example, some of the molecules in the SASP can stimulate the immune system, which is beneficial for fighting off infections. On the other hand, the SASP can also promote inflammation and damage to nearby tissues, which can contribute to the development of chronic diseases. Another way that cellular senescence can contribute to aging is by disrupting the process of tissue repair and regeneration. As we age, our bodies become less capable of repairing damaged tissues and organs, which can lead to a range of problems, including decreased mobility, increased risk of infection. and decreased cognitive function. Cellular senescence can contribute to these issues by disrupting the normal processes of tissue repair and regeneration. The potential benefits of cellular senescence. Despite the negative effects of cellular senescence, some researchers believe that it may have some potential benefits. For example, some studies have suggested that cellular senescence may play a role in preventing cancer. By preventing the growth and division of damaged or abnormal cells, cellular senescence can prevent the development of cancerous tumors. Additionally, some researchers believe that the SASP may have a protective effect on nearby tissues, reducing the risk of age-related diseases. In recent years, researchers have also become interested in the potential benefits of senolytic therapies. Senolytic therapies are treatments designed to target and eliminate senescent cells from the body. By removing these cells, senolytic therapies may be able to reduce the negative effects of cellular senescence and improve overall health and life expectancy. The future of cellular senescence research. Cellular senescence is a complex process that is still not fully understood. However, researchers are making significant progress in understanding the mechanisms behind this process and the potential benefits and drawbacks of senescence. In the coming years, we can expect to see more research exploring the links between cellular senescence and age-related diseases and developing new therapies to target cellular senescence. One area of research that is currently being explored is the impact of lifestyle factors on cellular senescence. Studies have shown that lifestyle factors, such as diet, exercise, and sleep, can impact the rate of cellular senescence. For example, a diet high in antioxidants may reduce oxidative stress and slow down cellular senescence. Similarly, regular exercise has been shown to reduce inflammation and delay the onset of cellular senescence. Additionally, researchers are also exploring the use of stem cells to reverse the effects of cellular senescence. As we age, the number of stem cells in our bodies decreases, which can contribute to the development of age-related diseases. By restoring the population of stem cells in our bodies, researchers may be able to prevent or reverse the effects of cellular senescence and delay the onset of age-related diseases. As our understanding of cellular senescence continues to grow, we may also be able to develop new ways to diagnose age-related diseases and track their progression. Currently, many age-related diseases are diagnosed based on symptoms alone, which can make it difficult to detect them in their early stages. By developing biomarkers that can detect the presence of cellular senescence, we may be able to identify individuals who are at risk of developing age-related diseases and start treatment earlier. However, it is important to remember that lifestyle factors also play a significant role in the development of age-related diseases. Making healthy choices, such as maintaining a balanced diet, engaging in regular exercise, and getting enough sleep, can all help to reduce the negative effects of cellular senescence and improve overall health and well-being. Ultimately, the study of cellular senescence has the potential to revolutionize our understanding of aging and age-related diseases. By continuing to explore this process and develop new therapies, we can work toward a future where older adults can enjoy better health and a higher quality of life for years to come. As the aging population continues to grow, research into cellular senescence and its impact on aging and age-related diseases will become even more important. By understanding the mechanisms behind cellular senescence and developing effective treatments, researchers may be able to improve the health and well-being of older adults and delay or prevent the onset of age-related diseases. Additionally, it is important for healthcare professionals and policymakers to take into account the impact of cellular senescence on the healthcare system. As the number of older adults increases, the prevalence of age-related diseases will also increase, leading to increased healthcare costs and potentially overwhelming the healthcare system. By investing in research into cellular senescence and developing preventative treatments, healthcare professionals and policymakers can help to mitigate these issues and ensure that older adults receive the care they need. We must also acknowledge the impact of socioeconomic factors on aging and age-related diseases. Older adults from disadvantaged backgrounds may have limited access to healthcare, healthy food, and opportunities for physical activity, which can increase their risk of developing age-related diseases. Addressing these social determinants of health is crucial for improving the health outcomes of older adults. Furthermore, it is important to note that the impact of cellular senescence on aging and age-related diseases may vary among individuals. Genetic factors, environmental exposures, and personal lifestyle choices can all influence the rate and progression of cellular senescence. Therefore, personalized medicine and individualized approaches to healthcare may be necessary to effectively target cellular senescence and prevent age-related diseases. Additionally, the impact of cellular senescence on mental health is also an important area of research. Age-related cognitive decline and neurodegenerative diseases, such as Alzheimer's disease, may be influenced by cellular senescence. A better understanding of the link between cellular senescence and mental health could lead to the development of new treatments and interventions to improve cognitive function and mental well-being in older adults.